In today's class, we are going to look at the effect of activators and inhibitors on enzyme activity. We are starting our introduction by looking at the active sites of enzymes. So first of all, what we have to note is that enzymes have active sites of specific shape as they have tertiary structures. And a small change in the size or shape of the active site can change the activity of the enzyme. Also, the active sites of enzymes can further be classified as the binding site, as seen here, and catalytic site, as seen over here. So what we have to note is that the binding site chooses the substrate and binds it to the active site, and the catalytic site performs the catalytic action of the enzyme in a reaction. Or we can say that the binding site of the active site of the enzyme holds substrate in place, whilst the catalytic site is the site where reaction occurs. Processes at the active sites. So from this illustration, we can see this is our enzyme, this is our substrate or the reactant, and this is the active site of the enzyme. So what you have to notice is that enzymes bind at the active site by covalent linkages with the substrate molecules to form an enzyme substrate complex, as seen over here, with very low activation energy and form products easily in a short time. And one thing we have to note again is that after the reaction, the enzyme release their product as shown here and come to their original form without any changes. Cofactors. Cofactors are the non-protein molecules that perform or catalyze reactions that are not carried out by the 20 amino acids present in our body. Cofactors can be organic or inorganic. Cofactors bind to the protein and make them active, as proteins without a cofactor are inactive. Promoters. Promoters are the chemical components that help in increasing the activity and efficiency of enzymes. Promoters are usually taken in a small quantity along with the catalyst in a chemical reaction. And what we also have to notice that cofactors and promoters are examples of enzyme activators. Enzyme inhibitors. Let us start by asking the question, what is an enzyme inhibitor? An enzyme inhibitor is a molecule that binds to an enzyme to decrease or slow down its activity. And what you also have to note is that enzyme inhibitors in some cases can stop the enzyme reaction or enzyme activity in an enzymatic reaction. Types of inhibitors. We have two types of enzyme inhibitors, and these are reversible inhibitors and irreversible inhibitors. The main difference between reversible inhibitors and irreversible inhibitors is that reversible inhibitors bind to enzymes by forming a non-covalent interactions or bonds, whilst irreversible inhibitors bind to the enzyme by forming covalent interactions or bonds. So this is the main difference between reversible inhibitors and irreversible inhibitors. We are looking at the types of reversible enzyme inhibition. So what you have to note is that we have three main types of reversible enzyme inhibition. And these are the competitive inhibition, the non-competitive inhibition, and the uncompetitive inhibition. So we are going to look at them one after the other. We'll start by looking at the competitive inhibition. So from this illustration, we can see this is our enzyme, this is the active site, this is the allosteric site, this is our substrate, this is our inhibitor. So first of all, what we have to note is that competitive inhibition is a type of inhibition where the inhibitor competes with the substrate for the same active site of the enzyme. Also, the inhibitor in this case is structurally and chemically similar to the substrate, hence able to bind to the active site of the enzyme to form an enzyme inhibitor complex as seen over here. And once an enzyme inhibitor complex is formed, it prevents the substrate from binding to the active site of the enzyme. Hence, there will be no reaction. And one important thing that we also have to note in competitive inhibition is that as the inhibitor is in competition with the substrate, its effects can be reduced by increasing the substrate concentrations. So I'm going to explain that using this second illustration over here. And this is the first part of this illustration, and this is the second part of this illustration. So from the first part of this illustration, we can see that at equal concentration of the substrate and the inhibitor is able to bind to the active site of the enzyme to form an enzyme inhibitor complex, as this prevent the substrate from binding to the active site of the enzyme. In the second part of this illustration, we can see that an increase in the substrate concentration will either reduce or stop the effect of the inhibitor in the enzymatic reaction. So in summary or in short, if you want to reduce the effect of the inhibitor in a competitive inhibition, we only have to increase the concentration of the substrate in the reaction. The next is uncompetitive inhibition. So from this illustration, this is our enzyme substrate complex over here, and this is our inhibitor, which is binding to our enzyme substrate complex. So in uncompetitive inhibition, the type of inhibition where the inhibitor binds to the enzyme substrate complex. So this is the enzyme substrate complex. And what you have to notice that the inhibitor can cause structural distortion of the enzyme active site, hence making the enzyme catalytically inactive. Also, unlike the competitive inhibition, uncompetitive inhibition cannot be reversed by increasing the substrate concentration, but rather an increase in substrate concentration will favor the inhibition concentration. This is because the inhibitor binds only to the enzyme substrate complex in uncompetitive inhibition. Non-competitive inhibition is a type of inhibition where the inhibitor binds to the enzyme at a location other than the active site. Or in other words, the enzyme binds to the allosteric site of the enzyme. Now let us look at this illustration over here. So this is our enzyme, this is the active site, and this is the allosteric site of the enzyme. This is our substrate, and this is our inhibitor. So what happens in a non-competitive inhibition is that the inhibitor binds to the allosteric site of the enzyme to form an enzyme inhibitor complex. And this changes the shape of the active site of the enzyme. So if you can see over here, looking at the shape of the active site of the enzyme and its shape over here, there is a change in the shape of the active site. And this, as a result, slows down or stops the reaction. Mixed inhibition. Mixed type enzyme inhibition is a form of enzyme inhibition where the inhibitor can bind to both the free enzyme and the enzyme substrate complex. This results in a distinct effect on both the enzyme catalytic activity and its affinity for the substrate. And one thing we have to note is that in mixed type inhibition, the inhibitor can interact with the enzyme at either the active or a separate allosteric site. So in summary, reversible enzyme, we have three main types of reversible enzyme inhibition. And these are the competitive inhibition, non-competitive inhibition, and uncompetitive inhibition. In the competitive inhibition, the inhibitor binds to the active site of the enzyme. In the non-competitive inhibition, the inhibitor binds to the allosteric site of the enzyme. And in uncompetitive inhibition, the inhibitor binds to the enzyme substrate complex, as seen in this diagram or in this illustration. Enzyme activators. So we will again ask the question, what are enzyme activators? Enzyme activators are molecules that bind to enzymes and increase their activity. These molecules are often involved in the allosteric regulation of enzymes in the control of metabolism. So examples of enzyme activators are cofactors and coenzymes. From the illustration below or from the picture below, the enzyme activator gets involved in the allosteric regulation of the enzyme by binding to the allosteric site of the enzyme to change the structural conformation of the enzyme, and in so doing changes the structure of the active site of the enzyme to increase its activity. So in summary, we can see that as the activator binds to the allosteric site of the enzyme here, it changes the shape of the active site from this to this. As the new shape of the active site allows the substrate to bind to it more easily, and as a result increases the enzyme activity. Enzyme activators include ions, small organic molecules, proteins, or lipids. Moreover, many enzymes contain specific sites for the binding of small inorganic ions, especially cations such as calcium ions. Here, the binding of these ions changes the conformation of the enzyme molecule to activate it. Therefore, these ions serve as cofactors. And what we also have to notice is that cations such as magnesium ion binds to the substrate to decrease the negative charge of the substrate to facilitate the binding of the substrate to the enzyme. Also, some of the heavy metal cations eliminate inhibitors. On the other hand, some of the chelating agents, including EDTA and EGTA, bind to the inhibitory cations to eliminate the inhibitory actions. Some enzymes activators like camodulin, which is a calcium binding protein, also form a complex with the target enzyme to activate the enzyme. Also, fructose 2,6-bisphosphate is a small organic molecule which serves as an enzyme activator whilst increasing the rate of glycolysis. Also, hexokinase 1 and glucokinase are proteins which activate enzymes. Allosteric site. So first of all, let us just study this enzyme over here. So this is an enzyme with it, and this happens to be the allosteric site or the regulatory site of the enzyme, and this is the active site or the catalytic site of the enzyme where the substrate binds to. And this is our allosteric effector, which can either be an activator or an inhibitor, binding to the allosteric site of the enzyme. So now, what you have to note is that the word allosteric comes from the Greek language and translates to the other side. And what you have to note is that allosteric sites are effector binding sites within enzyme, distinct from the active sites of the enzyme. So this happens to be the allosteric site of this enzyme, and it is where the allosteric effector binds to. An effector is any molecule that binds to the enzyme that is not the substrate of the active site and modifies the enzyme activity. So as you can see, this is an effector which is binding to the allosteric site of this enzyme and not the active site. Effectors bind to the allosteric site of an enzyme to modify the activity of the active site. If effectors can increase or decrease the activity of the enzyme they bind to and may be reversible or irreversible. Allosteric binding. So what you have to note is that allosteric binding is the process of an effector binding to an enzyme to modify its activity. The binding of an effector to an enzyme changes the structural conformation of the enzyme, and in doing so, changes the structure of the active site. Elements such as magnesium or calcium, small molecules including CGMP and ADP, and proteins can be effectors. And one key thing that we have to notice is that changing the structural conformation of an enzyme through an effector binding to the allosteric site can modify the active site to be more active. For example, when CMP binds to N3 motif in protein kinase A and not the active site, the protein kinase A activity is increased. And what we also have to note is that effectors can also modify the conformation of the active site to decrease enzyme activity. Allosteric activator. An allosteric activator is any molecule that binds to an enzyme allosteric site and increases the activity of the enzyme. Allosteric activators enable the rapid increase of enzymatic activity, thereby increasing the overall activity and efficiency of the enzyme. So these are just some questions and answers that I would just like you to take time and just go through. So the first says, how do allosteric activators bind? The second question says, what happens in the allosteric site? The third, what is meant by allosteric effect? And the last question, what happens when an enzyme is bound to an allosteric activator? So I would just encourage you to go through these questions and answers. Membrane permeability and dialysis tubing. So first of all, this is how the dialysis tubing looks like. So let's see what dialysis tubing is. Dialysis tubing is a selectively semi-permeable membrane, usually made of cellulose acetate. It is used in dialysis, a process which involves the removal of very small molecular weight solutes from a solution along with equilibrating the solution in a buffer. Dialysis is often used in diffusion demonstration. Membrane permeability. A selectively semi-permeable membrane is a cell membrane that permits some materials to pass through and some not to pass through. Usually, molecules smaller than the pores in the membrane are able to pass through, whilst those larger than the membrane pores are retained. So as we can see from here, these are molecules smaller than the pores of the membrane. So they are able to pass through the membrane. And these are molecules that are larger in size than the pores of the membrane. So they are retained in the dialysis tubing. Also, the molecules or materials diffuse from an area of a higher concentration to an area of a lower concentration. So what it means is that the molecules in the dialysis tubing is of a higher concentration as compared to the solvent in the beaker over here. So from this picture or diagram over here, this is our selectively semi-permeable membrane tied at both ends. This is one end and this is the other end, and dipped in a solvent of a lower concentration, say distilled water in a beaker. Also, the molecules in the dialysis tube is of a higher concentration, but with different molecular sizes or molecular weight. So as we can see, so this molecule has a higher molecular weight than this molecule over here. And what you have to note is that the larger molecular weight molecules are not able to pass through the pores of the membrane, therefore are retained in the dialysis tube after dialysis. On the other hand, the smaller molecular weight molecules have sizes smaller than the pores of the membrane, therefore are able to pass through the membrane pores into the solvent in the beaker to equilibrate it as shown here. Dialysis. So first of all, what we have to note is that to dialyze means to separate or remove low molecular weight substances which are present in a solution of higher molecular weight compound by allowing the smaller molecules to diffuse through the pores of the sealed dialysis tubing into a larger volume of solvent. The enzyme salivary amylase. So what you have to notice is that saliva is a thick, colorless, opacent fluid that is constantly present in the mouth of humans and other vertebrates and produced by a salivary gland in the mouth. The saliva is composed of 99% water and 1% protein and salt. The predominant protein is the alpha amylase or the thylline. The other proteins, which are glycoproteins, are immunoglobulins, cystathins, proline-rich glycoprotein, the salivary agglutinin, lysozyme, cystatins, histatins, lactoferrin, and mucin are modified forms of protein produced by glycosylation, which is a process by which a carbohydrate is attached to a protein. The mineral salts are sodium, magnesium, bicarbonate, phosphate, potassium, calcium, and chloride, and these salts act as electrolytes. How to dialyze saliva. One key thing that we have to note is that the process is carried out in a refrigerator. So to dialyze a saliva, you place an amount of saliva about 15 to 20 mils in a dialysis tube of length 6 to 8 inches. You tie both ends as seen over here and place in a beaker containing distilled water by suspending from a glass rod. So when you are done with this, you place in a refrigerator and change the water every day. That is every morning and evening for about 3 to 5 days. So let's see what happens at the start of dialysis and also what happens at the end of dialysis. So the setup A shows the start of dialysis and setup B shows how the smaller molecular weight molecules leaves the membrane through the pores to the solvent in the beaker to equilibrate it at the end of dialysis. Importance of dialyzing saliva. The importance of dialyzing saliva is to separate and purify the enzyme salivary amylase by removing other components in the saliva through diffusion. Reagents and materials needed for the test protocols. So the first is our 2% starch solution that needs to be freshly prepared, iodine solutions, white tile, 0.01 molar phosphate buffer of pH 7.0, 0.2 molar solution of sodium chloride, potassium chloride, magnesium chloride, and potassium bromide, and pasture pipette. The first step in the test protocol is substrate buffer preparation. We prepare our substrate buffer by adding 2.5 mils of the 0.01 molar phosphate buffer of pH 7.0 to 2.5 mils of the 2% starch. The second step is the equilibration of the enzyme and substrate buffer at 37 degrees Celsius for 5 minutes. And this is done by putting the 5 mil substrate buffer and the dialyzed enzyme in different test tubes into the water bath set at 37 degrees Celsius and allowing them to stand for 5 minutes to equilibrate them. The third step is reaction mixture. We perform the reaction mixture by adding 0.25 mils of the equilibrated enzyme to our 5 mil substrate buffer in a water bath set at 37 degrees Celsius. And what we also have to note is that we have to perform a control for the reaction mixture. And to perform a control for the reaction mixture, we add 5 mils of distilled water to the reaction mixture and then subject it to the iodine starch test to check when the reaction will be complete. Also, to check for the effect of activators or inhibitors on the reaction mixture, we add 5 mils of salt to the reaction mixture and then subject it to iodine starch test. And what we have to note is that we have 4 different salts over here. So for each salt, we do a separate setup for each salt. So what it means is that for sodium hydroxide, you have a setup. For potassium chloride, we also have a different setup. For magnesium chloride, we will have a different setup. And for the potassium bromide, we also have a different setup. The fourth step is the performing of the iodine starch test. For the control, we do this by spotting a drop of the reaction mixture onto a white tile containing a drop of iodine. And we repeat this every minute until the reaction is complete. And the reaction becomes complete when the blue-black color formed at the initial stage turns completely to yellow or start to produce a yellow color in sequence. So from our test result over here, we can see that the reaction is complete for our control at 8 minutes, because at 8 minutes, the reaction turns yellow or start to produce a yellow color in sequence. Also, for our salt, we perform the iodine starch test by spotting a drop of the reaction mixture containing the salt onto a white tile containing a drop of iodine, and we repeat this every minute until the reaction is complete. And the reaction still becomes complete when the blue-black color formed at the initial stage turns completely to yellow or start to produce a yellow color in sequence. So from this test result, we can see that the reaction is complete at 5 minutes for our salt sodium chloride. So we should also note that if reaction is too fast, we dilute the enzyme. Example, we can do a dilution of 1 in 10 or 1 in 20 or 1 in 50 or 1 in 100. And if reaction is too slow, we increase the volume of the enzyme. Let's see the test result. So this is the table of results showing the time taken for complete reaction to occur in minutes for the control and each of the salt. So we can see that for the control, the time taken for a complete reaction is 8 minutes. For the sodium chloride, it's 5 minutes. For the potassium chloride, it's 6 minutes. For the potassium bromide, it's 7 minutes. And for the magnesium chloride, it's 15 minutes. So now let us look at the calculation. So rate of reaction is calculated as 1 over time, which is the inverse of the time taken for a complete reaction. And the table of results for the ions, time taken for a complete reaction, and rate of reaction is shown here. So we can see for the control, the time taken for a complete reaction is 8 minutes. And the calculated rate is 0.125 per minute. That of sodium chloride is 5 minutes. And the calculated rate of reaction is 0.2 per minute. That of potassium chloride is 6 minutes. And the calculated rate is 0.167 per minute. The potassium bromide is 7 minutes. And the calculated rate is 0.143. And that of the magnesium chloride is 15 minutes. And calculated rate of reaction is 0.067 per minute. So the next thing to do is to plot a graph of the rate of reaction per minute against our ions. And the nature of graph that we are going to plot is a bar graph. So this is the nature of graph that we are expecting to get. So this is for sodium chloride, potassium chloride, potassium bromide, our control, and our magnesium chloride over here. And from theory, what we have to note is that activators speed up the rate of enzymatic activity, while inhibitors decrease the rate of enzymatic activity. And using the distilled water as a control, we can see that the rate of the enzymatic activity for the reaction mixture containing sodium chloride, potassium chloride, and potassium bromide has increased, whereas that for magnesium chloride is reduced or has decreased. Therefore, we can conclude that the sodium chloride, potassium chloride, and the potassium bromide are activators, whereas the magnesium chloride is an inhibitor. Thank you.